Okay, good afternoon, everyone. Welcome back. Today, we're going to carry on our discussion about bacterial disease, specifically on human beings, but we're going to find, especially today, that this touches also on bacterial populations of other organisms as well. This is one of the places where we're going to start intersecting with other animal populations. Today, we're going to be discussing microbial disease of the digestive system, amongst the most important of all human diseases, because these diseases continue to exert a very, very strong pressure on the human population. There's a very high human mortality annually due to diarrheal diseases in particular. And these exert the particular effect on young children and on infants. Other people as well, it can be, but the infants and young children are especially vulnerable to diarrheal disease because the diarrhea itself exerts such a strong effect on the small body. The volume of fluids lost can be very high, proportional to the total volume that the infant has. So they can suffer ill effects from diarrhea very, very quickly. So before we begin, let's just very quickly, this is not an anatomy lesson, but we just want to remind ourselves about different parts of the digestive system and one or two of the characteristics, because we're going to be hearing about them again. First of all, obviously we eat through our mouths, and there's oral cavity, tongue, and all the attendant tissues of the mouth, all invested with a mucous membrane, and lubricated also by the salivary glands. The mouth, it connects to the stomach via the esophagus. The stomach itself has its own environmental characteristics. It has a very low pH. It leads to the small intestine, where the pH is rather slightly more than neutral. It's a little bit alkali. The large intestine follows on the small intestine, and then, of course, is evacuated. This is a tube all the way through. So here's the thing that I want to impress on you. When we look at a diagram like this, we see that the digestive system runs from the mouth to the anus as a tube. So all of the way through here, we are passing material, and we also have the possibility of building up bacterial populations. The gut wall is literally a wall. It is a barrier between this passageway here and the interior of the body in the abdomen, the peritoneal cavity, etc. The interior of the body here is kept largely sterile, but the entire digestive system is populated by its own bacterial populations, which are completely natural and in fact which are essential to the functioning of the digestive system. If we look at the mouth has a very rich bacterial flora. Thousands of different species normally occur there, and they occur in complete balance with the host, with the human being. The esophagus also has a microflora, not as rich as the mouth. The stomach has a very, very low bacterial population, largely because of the pH. The pH is too low for most bacteria to survive. As soon as fluids leave the stomach, however, they are neutralized. Acid is neutralized by pancreatic juices. And the small intestine has a very rich microflora. The large intestine has a huge microflora. So all the way through, we're dealing with completely natural microbial populations, which in their normal situation remain isolated to the gut cavity. But it's really important to realize, even these benign commensal bacteria that we have in our gut, if they were to move across into this otherwise sterile cavity that forms the cavity of the body cavity, that immediately can cause huge problems. Likewise, if any of these bacteria start to enter the bloodstream, they can cause huge problems. There are very strong reactions against them. So everything is fine as long as the populations are well regulated, the populations are balanced, and the bacterial populations stay where they ought to be. Then everything works fine. We're going to be talking about what happens when some of these mechanisms go wrong. So most should not surprise you to know that the body devotes a huge amount of energy to maintenance of the immune system of the gut, because gut is the one place where we are continually exposed to materials from outside. The immune system reacts to antigens. Those antigens can be food, or it could be infectious particles or whatever. The antigens are things which excite the attention of the immune system. And we are continually exposed through eating to a rich array of different things, different organisms, different substances, etc., which could actually be damaging to the body. And a lot of the immune system lies along the gut cavity. In fact, about 70% of the immune system, if we look at it just in tissue-wise. There are very complex systems. One of them you've already heard of. This is MALT, mucosal associated lymphoid tissues. We talked about that when we talked about airborne disease, because those are the parts of the immune system which are associated with mucosal surfaces, and they are very important in respiratory surfaces. That is true, but they are also important in the gut cavity as well. But there is another set of immune tissues called GALT, gut-associated lymphoid tissues, sorry. And when we go through the whole anatomy of the gut, these are actually visible. And there is a rich investment of lymph nodes, for example, all the way along the gut. And there are also in the small intestine, there are specialized areas called Peyer's patches, which are kind of like flattened lymph nodes, if you want to think of it like that. All of these devoted to surveillance of the material which is passing through the gut. There are also some cells which we need to remember because they become very important in some pathogenic situations. And these are called the microfold or M cells. And what they do, they're an extremely interesting set of cells which sample material continuously as it passes by. And if it is antigenic, they alert the rest of the immune system and set up an immune response. Then there are a whole lot of cells which are purely protective. One lot called the Paneth cells, and they produce antimicrobial granules specifically for pathogens, antipathogenic. And then there's a whole population of phagocytic cells which meander through the gut continuously and produce, and not only sample and eat potential pathogens, but which also produce proteins, things which are antimicrobial if they encounter something which is potentially pathogenic. So the take-home message is that the gut is guarded by a specialized immune system, its own sort of immune system. But probably the greatest protection which we are offered is not necessarily from the immune system, which reacts to the presence of pathogen, but from the presence of our own microbiota. We have a hugely diverse microbiota through the gut. Thousands upon thousands of species continue to be discovered as this is investigated. I've already told you the microflora of the mouth is very rich. That of the stomach is rather poor. The small intestine has its own specialized microbiota, and large intestine, which doesn't perform much digestive function apart from absorption of water, consolidation of the feces, actually it cultivates a huge population of bacteria, about 100 million bacteria per gram of feces. So the first disease process that we're going to talk about is actually in the mouth. And it's something which in modern times is probably far less of a problem than it used to be. Nonetheless, it still carries with it a considerable morbidity. It causes illness and distress to many, many people. And these are diseases of the mouth, especially of the teeth. And it's unsurprising that the teeth should be a problem because they're actually extremely complex structures. And they represent a boundary, if you to think of it like that. Part of them is outside, exposed to this rich microflora and whatever else. Part of them is embedded within the skeleton. So they can potentially provide a gateway from the outside to the interior of the body. Now, under normal circumstances, this very rich microbiota of the mouth keeps everything in balance. The teeth do not need to be kept completely clean of bacteria in order to remain healthy. What they need is to have a healthy microflora around them. So here's the basic structure of the tooth. I'm not going to go into it in any detail, but this is what I mean. Here's the top of the tooth, the functional part of the tooth, which does the biting and the chewing and everything else, exposed to the outside. Here's the interior of the tooth, actually embedded in the skeleton, embedded in the blood vessel-rich skeleton. The tooth has its own anatomy. It has its own living tissues inside, shrouded by, first of all, by relatively soft bony material called dentine, and the dentine shrouded again over the top by very, very strong enamel. So under normal circumstances, the tooth is kept sealed. The roots of the tooth are kept sealed from the outside. The microflora would occupy the outside here of the tooth, okay? And under normal circumstances, that is the way it persists. There is certainly evidence that as you age, there are inevitable changes that are going to take place to the tooth, especially because teeth can get worn down and expose the softer dentine, et cetera, et cetera. But under normal circumstances, even very, very old people can maintain healthy teeth if their diet is healthy. And here, that's the major thing which affects the structure and the functioning of the teeth. The reason for this is that as diet changes, so the microflora changes. And in particular, we change the nature of the biofilm which covers the tooth. Normally, that biofilm is protective. If you want to think about it, it only cultivates healthy bacteria. Especially if we start to take in a diet which is low in fiber and high in refined or sugars, things like that, we begin to alter the structure of the biofilm. And especially this organism here has been implicated. And this is Streptococcus mutans. And Streptococcus mutans, here it is here. It's a gram-positive, a facultative anaerobe occurring in typical Streptococcus chains. But it has this following characteristics that make it very dangerous in the mouth. And that is, it produces external substances called dextrans. They're polysaccharides, which form a very tough layer, which begins to form a biofilm with the Streptococcus mutans involved in it, but it also binds and attracts other bacteria which can cause tooth decay. And Streptococcus mutans is typical because what it does is it takes these refined sugars, like ordinary table sugar or whatever you're eating, sweets and candies and all the rest of it. It takes those sugars and it converts them to lactic acid, which is released and begins to eat its way through this enamel there. So that is the first impact. The second impact is it begins to affect the populations of bacteria here, in between the teeth, and especially in these crevices here. And now, instead of healthy bacterial populations there, to which the body does not react, we begin to build up populations of bacteria which actually cause inflammation of the gums. And so now the process of tooth decay in general is involving two processes. The first is that we're beginning to affect the structure of the enamel and beginning to get holes in the teeth. Those holes potentially can make their way right the way through the enamel into the dentine, and through the dentine into the pulp, into the living center of the tooth where the blood vessels and nerves and everything are. So that is the one set of disease processes, is the actual decay of the tooth. But also happening at the same time, there are changes which take place in the gums surrounding the teeth. And when you look at those little cavities and you change the populations there, what begins to happen is, first of all, those biofilms begin to build up and start to penetrate between the gum and the tooth. At the same time, the bacterial populations set up an inflammatory process in the gums. And the gums, in fact, begin to recede. They begin to move away from the surfaces that they are usually closely applied to. Bacteria build up between them, and we begin to form pockets of bacterial-rich material lying against the tooth, further enhancing the inflammation. This initial inflammation here, this is called gingivitis, inflammation of the gums. It's easily visible often as inflamed gum tops of the gums, especially here at the top where you can see into the crevices between the teeth. And you may also notice that this is when your gums begin to bleed very easily. This is the first sign of gingivitis. Gingivitis left alone, these processes, these bacterial populations along the edges of the tooth become incredibly dense, and the gums retreat even further. And eventually, they retreat so far down. Let me get back to this. They retreat so far down that these soft areas of the tooth are exposed and very easily begin to decay. And simultaneously, the bone here begins to recede. So in fact, the whole support structure for the tooth loosens, and the tooth can actually be lost. And so these different stages, this is gingivitis, the very start, the start of the inflammation, the establishment of these populations between the gum and the tooth. Periodontitis is the retreat of the gums completely, and the exposure and retreat of the bone, supporting bone material itself. These are very dangerous situations. And you may be surprised to know that in ancient times, tooth decay and periodontitis were major mortality factors in human populations. Many of the, for example, Egyptian mummies that have been shown that many of them died of ulcerative and infectious processes which arose because of their poor dental hygiene. Okay, so brush your teeth. Well, should we? Here's a question. Is it actually better to brush our teeth? is it a good idea to remove all these bacteria with dental floss and with mouthwash and everything else? Or would it be better to pay more attention to our diet and try and nurse a healthy microbial population in the mouth, which would protect us far better? That's a question you might want to think about. Okay, so that's a quick bit of dentistry for the day. But don't forget these dental problems are a massive drain on the workforce. And for many people, especially in poorer areas, people who don't have ready access to good dental care, they can be actually quite dangerous as well. All right, so let's talk now about the actual disease processes caused by bacteria coming in from outside, by bacterial processes that originate outside. And the very first one I'm going to give you is an example of a toxicosis. And a toxicosis is a disease process where the person suffers disease because of the presence of a toxin, not because of the presence of the bacterium, but because of the presence of toxins which the bacterium releases. We've already heard about this when we talked about respiratory diseases. Some of those respiratory bacteria cause disease by release of a toxin, not by just by their presence. So when we talk about the gut, we talk about these toxins, especially the one really important group are the enterotoxins. And the enterotoxins are those bacteria, those substances which are released as an exotoxin, which cause intestinal distress. So an enterotoxin causes vomiting, it causes diarrhea. Those are the principal effects. Whenever you hear me say enterotoxin, just remember it's an exotoxin. It's released by the bacterium. And here is a typical one. It's our old friend, Staphylococcus aureus. And Staphylococcus aureus is this clustered coccus. And this is, in fact, a common member of our skin flora. And unfortunately, it also frequently will set up small pockets of infection on our skin. This is free. You get an infected cut, or if you cut your cuticle and it gets a bit infected, often it's an infection with Staphylococcus aureus. This becomes really significant because your little cut that's a bit infected, it may be painful. If it persists, you'd probably go and get some antibiotic to treat it, etc. But it's external to you, effectively. It's on your finger, it's in your skin. But just picture this, that you're a food worker, and you're preparing foods, you're preparing egg salad, or you're preparing something like that, and you have a small cut which is infected on your finger. You may hardly even notice. But if you're not careful about your hygiene, if you're not wearing gloves or whatever, you can infect that food with Staphylococcus aureus from your cut, from your infection. This is surprisingly frequent and is a common cause of food poisoning, which arises from commercially prepared foods where food preparation hygiene has not been carefully observed. And what happens is that the bacteria, Staphylococcus aureus, reproduces very, very quickly. So it builds up population inside the food item very quickly. It's not the presence of the bacteria. In fact, the bacteria will be killed when you swallow the food. They'll mostly be killed by your stomach acids. But the whole time that they're growing inside the food, they are releasing this enterotoxin, and the concentration of enterotoxin builds up in the food. And then people eat the food, and within a half an hour to the most a couple of hours, the enterotoxins cause violent diarrhea, nausea, vomiting, etc. This illness is because of an intoxication, not because you've been infected with the bacteria. The bacteria can long be dead. But unfortunately, there's a couple of things about Staph aureus. The first is, even if the food is heavily salted, Staph aureus doesn't care. It grows on your skin. It's resistant to high salt content where many other bacteria would not survive. Second of all, that food, especially if it's put onto a hot tray or something like that, they just love it. And they grow at a really high temp. They'll grow at a really high temperature. And even if you cook the food afterwards, if they have had a period of time to grow and release the exotoxin is not affected by heat. You can kill the bacteria by heat if you want to, but the toxin stays. Now, this is the typical kind of church picnic. I always think of food poisoning, where somebody has prepared their fruit, their egg salad or their potato salad or whatever, put it on, they've infected it with Staph aureus from a small cut or something like that. They put it onto the side table, and while everybody's gossiping and doing whatever, then 3 hours later they come back to eat, and they eat the potato salad, and within a half an hour everybody's running for the bathrooms. This is a typical situation also on airplanes, these sorts of things, and its hallmark is that the symptoms develop very, very rapidly after eating, because you don't have to have these things growing inside your gut to be sick. You just have to eat the poison. Normally completely self-limiting. In very old people or in children, if it persisted, you would want it. But for most people, it's just a huge inconvenience. This is another one. This is shigellosis, and it's caused by this gram-negative bacterium called Shigella. And Shigella is actually a bacterium which is quite closely related to many of the other gut bacteria. The problem with it, though, is there are many strains of Shigella, and it's a facultative anaerobe. It's gram-negative, and there are many different types of Shigella. Some of them completely non-pathogenic, others which cause a very, very severe illness, and they have some interesting mechanisms which we need to pay attention to. The first thing is that some of these strains of Shigella produce a very, very powerful enterotoxin. It causes really catastrophic, devastating diarrhea and vomiting. But accompanying that is the fact that Shigella is actually growing inside the gut. This is an infective process. And the Shigella actually causes damage to the gut wall. And that damage can be extremely serious. The Shigella produces a dysentery. That is, it produces a violent diarrhea with blood, with bleeding, and often with bloody mucus as a product. In addition, some of these strains of Shigella produce a very, very serious condition called hemolytic uremic syndrome. This is from the breakdown if they enter the bloodstream, and they cause the breakdown of hemoglobin, and the hemoglobin products are toxic to the kidneys and cause a very, very severe inflammatory condition of the kidneys. So these Shigella can be extremely serious. A Shigella infection can in fact be fatal. Unfortunately, Shigella is one of the organisms which survives passage through the stomach quite well. So you only need a relatively small dose. And here is our very first instance, very common to all of these gut bacterial disease. Shigella is usually acquired through fecal oral contamination. So it is from fecal oral transfer, food or water contaminated by feces from people who already have the disease. it begins to, as the disease moves through the gut, the population of Shigella is enormously increased. So if people then defecate near a water source, for example, or if they use poor hygiene and contaminate food with their hands, they can transfer an enormously high dose of Shigella, which can be acquired by other people swallowing it. So here's what I meant by the mechanism. Shigella has this very strange way of finding these M cells. And you remember I told you about these. These are cells which actually are specially designed to continuously sample material from the outside to inform the immune system if pathogens are present. What happens here is that the Shigella actually induces M cells to endocytose them. They go into the cell and they survive and begin to breed inside the M cells. And they then are able to move through the tight junctions that tie these cells to cells on either side. They begin to migrate out into the intestinal cells on either side. So you can picture it like a colony growing out from their initial point of infection. They are very damaging. They're very cytopathic to the cells, and the cells begin to break down. And this is the point at which Shigella can actually begin to enter the bloodstream and become disseminated. The spreading into the bloodstream is not that common, but it can happen. But the formation of plaques of intensely damaged and inflamed tissues along the gut wall, these are the things which produce this in very, very debilitating cramping and bloody dysentery of the gut wall. Shigella is a very serious, it can be a very serious infection. It does depend on the strain. There are very often, it's relatively self-limiting, even without antibiotic treatment or whatever. But if it persists and the presence of Shigella is proved by testing, then usually it would be treated with antibiotics. Now, this mechanism here, this ability to move into the intestine is conferred by a virulence factor. And it's actually a set of toxins called the Shiga toxins. And this is important because we begin to see these similar mechanisms in other organisms that cause these diarrheal diseases. And the next one is one that's surely familiar to you. And this is salmonellosis, I'm sorry. And salmonellosis is caused by this organism called Salmonella. And Salmonella is actually a kind of a catch-all name for a very big group of organisms, of related bacteria. There are more than 2,000 different serotypes have been recorded. That means that at least their exteriors look different to our immune system, right? There are 2,000 different ways that these organisms can cause a reaction in the immune system if we test for them. They vary tremendously in their pathogenicity. Some of them are completely non-pathogenic and just part of the normal gut flora. Others are very, very pathogenic and cause extremely serious disease. And then there's a huge number which are kind of in between, which cause diarrheal diseases, which are particular importance because they're very often foodborne. And again, it's very often fecal oral contamination. It's a gram-negative facultative anaerobe, rather similar to E. coli in many, many ways. And if you're dealing with a strain of Salmonella, which has the particular virulence factors, they begin to behave rather like Shigella, and they begin to reproduce in M cells and spread out. That's the first thing. But they are also able to infect macrophages. Now, this, again, disease from Salmonella is because of ingestion of living cells, which then populate the gut. They certainly do produce exotoxins, but you need growing Salmonella population in order to fall ill. So there's a longer incubation time, 12 to 36 hours. But Salmonella, I'm sure some of you had it, but you will know it causes very profound nausea, gut cramping and diarrhea, but also a fever, which is not always a characteristic of these diarrheal diseases, but Salmonella very often does. So it's very, very common. And one of the reasons for that is it's very often associated with things that we use as food. So first of all, you'll see, look at the 1 million cases in the United States per year, only 380 deaths though. So while salmonellosis, Salmonella gastroenteritis is very common, it has a low mortality rate, especially nowadays. And in general, the treatment is to support the person who is ill, to limit pain, try and limit the diarrhea without stopping the diarrhea, because you don't want the populations of bacteria building up in the gut. And then watching to see, watchful waiting is often the byword. Right up until the 1980s, 70s, 1980s, they would slam the person full of antibiotics. But that is now not usually done. Usually you would try and avoid the use of antibiotics because you don't want to disturb the normal gut flora. You want the gut flora to return to its normal situation once the Salmonella has been avoided, which it usually will be eventually. The organism itself is very commonly associated with poultry and with reptiles. This is unfortunate. In fact, it can build up very high populations in bird guts, and not only birds that we eat, like chicken or duck or whatever, but also in pigeons and things like that. And so there is a very big population of potentially pathogenic salmonellas out there the whole time. And many, many, many of the exposures to Salmonella occur through poor meat husbandry. So for example, going to a factory system where chickens are being killed. If they are not very, very careful about keeping their line scrupulously clean so that they are never contaminated by chicken feces, it is possible to contaminate meats with feces and have Salmonella contamination of meat, which if that meat is eaten undercooked especially, can cause infection with Salmonella. The other common source of salmonellosis is from eggs, because eggs are often contaminated the outside with chicken feces, which may have Salmonella present. Salmonella survives dehydration quite well. So in addition, Salmonella can penetrate into eggs through the shell and survive there. The symptoms of salmonellosis, of mild salmonellosis, actually mimic many other diarrheal diseases. So that's if the disease persists, if the symptoms persist for more than a few days, usually you will do a fecal test to see if in fact you're dealing with Salmonella and what strain of Salmonella is being dealt with. Otherwise, treatment is usually supportive. Now, that is normal salmonellosis. And that's the one to which any of us might be exposed. I should issue one word of warning to you as well. Those of you with reptiles, those of you who have pets, iguanas or snakes or whatever, be very, very careful about your hygiene, about your hand hygiene. You should wash your hands assiduously after you've been handling either the animals themselves or stuff from their cages or whatever. Because very, very commonly reptiles maintain very high populations of salmonella in their gut. But with good hand hygiene, it's not a problem. But don't go kissing your iguana. So here, though, is another strain of salmonella. It's difficult to say that this is a different species because it's difficult to say in bacteria what is a species and what is not. But this is a very distinct serotype of salmonella. Probably it's just Salmonella enterica, but it used to be called Salmonella typhi or Salmonella paratyphi. And that is because it produces a very distinct disease syndrome. And this is called typhoid fever. Now, this is not to be confused with the disease typhus. Typhus is a disease which is caused by a very small kind of bacteria called a rickettsia, and is transmitted by ticks and fleas and things like that. But typhoid fever is a diarrheal disease caused by salmonella. And it's a very specific, it is a specifically human disease. We cannot, not like other salmonellas that we can acquire from animals. Salmonella typhi is specific to humans and it has to be transmitted human to human. So it is transmitted by human feces. Unfortunately, we only need a very small dose of the bacteria in order to establish an infection. So it is actually quite easily transmitted if there is poor hygiene. In the old days, when people just depended on pit latrines or whatever, this was actually far more common, and it was had major impact on human populations. Death from typhoid fever right up until the 1920s, 1930s was incredibly common, and it can occur at any age. So at the start, it looks much like any salmonellosis. You get a gastroenteritis, vomiting, maybe nausea, fever, but surprisingly enough, it is not usually accompanied by a diarrhea. And the bacteria, unfortunately, do not remain isolated to the gut. Instead, they begin to migrate through the body. This is where the fever part comes in, because they begin to cause a very profound reaction from the immune system. As the bacteria spread, they, of course, in the course of the population growth, etc., they begin to die. And as they die, the particles, remember this is a gram negative, the particles, especially of the outer membrane, go back and revise your bacterial anatomy. Remember what the outer membrane is. As those outer membrane components begin circulating, the immune system reacts extremely strongly to them, and people go into this condition called septic shock, which is a catastrophic loss of blood pressure in response to the presence of these gram-negative components. And they also begin to suffer from organ failure due to all sorts of circulatory problems as well, all the result of the immune response. So this can be a very serious situation. And the other thing that happens with typhoid fever is that the bacteria are actually able to cause complete necrosis of areas of the gut wall. And once that perforation occurs, then you get peritonitis and you get a spread of bacteria throughout the body and a septicemia. So in the old days, this was a critical medical situation. typhoid fever had a very high mortality rate. And many of the ways, I don't know if I've got a finger of it, no, I don't. But many of the effects of typhoid fever are again through those Shigella-like mechanisms of the bacteria invading intestinal tissue and then moving out and causing tissue damage and then entering the blood system. Nowadays, typhoid fever is actually rather uncommon in most areas of the world. The first reason is because we pay so much attention now to sanitation and to hygiene. These simple things that we do, like washing our hands after you've been to the bathroom, those sort of things, actually have an enormous impact in terms of population. So in addition, in cases where typhoid fever is suspected, it is very easily tested for by testing feces and characterizing the bacteria. And it can be treated with antibiotics. There is one major problem with typhoid fever, and this is particularly true, particularly important in the 19th century, 20th century, when people were incredibly mobile and people were moving from one continent to another continent. And that is that some people, when they recover from an infection, even a mild infection with Salmonella typhi, they can actually become carriers. They maintain a population of bacteria in their gallbladders, surprisingly enough. And they no longer suffer from any ill effects, but they are continually shedding bacteria, and they become a source of infection. And the most famous one of all was right here in New York, was a woman who became known as Typhoid Mary. Typhoid Mary worked right here on Long Island in Brooklyn, and she was a housekeeper. And unfortunately, she was a housekeeper with poor hygiene. And the households where she worked were singularly susceptible to typhoid fever. And many of the areas houses where she worked, the people suffered and died of typhoid fever. She was eventually identified, and she actually went into hiding. She knew that people were dying behind her. She carried on working and never improved her hand hygiene. Remember that in those days, this was in the 19th century, households mostly had pit latrines in the basement. They didn't have, even upper middle class families did not necessarily have running sewage. And so she was cooking the family meals, etc., and what she was doing was infecting them. Eventually, she was identified in court, and she was effectively imprisoned on an island in the New York Delta for the rest of her life, to keep her away from situations where she might infect people. Okay, so this is an interesting graph, because this is the incidence of salmonellosis and of typhoid fever. There's something I just want to show you. One thing is very easy to understand. That is the decline in the incidence of typhoid fever. It's now incredibly low. It's a rare disease in the United States and in Europe. It's not that rare in developing countries, especially where sanitation is not that good. So this decline in the incidence we can understand quite easily. What is a little bit more difficult to understand is this here. Look at the incidence of salmonellosis. It is increased between from 1934 right up until the 1980s. It was on the increase. So let's just think about that for a minute. What might lead to, it's almost an exponential increase. What might lead to that increase in salmonellosis? Well, there's a couple of answers. The first is that we're testing for it. Testing is getting better and better. Testing is getting easier and easier. So we're going to find more. Okay, we're going to find many people here would have simply wouldn't have done anything about it. They would have probably in the old days simply have gone, they would have, you know, just weathered the storm and not even gone to a doctor or whatever until they became extremely ill. This is measuring the incidence of salmonellosis in general. The second thing is that what you're also contributing here is the impact of commercial food production. So we are getting more and more, we don't grow our own food anymore. We don't buy our food locally. We're buying from factory farms. We're buying eggs. We're probably eating more eggs than people ever ate in the world ever before. We're exposing ourselves to likely sources of salmonellosis far more. From it seems to have peaked around about the 1980s, and now it's pretty much settled down to a background level there. But you can see that typhoid now, as a component of salmonellosis, of salmonella illness, is now very small. Okay, so here's another of these bacterial diseases which have had an enormous impact on human population. This is the disease of cholera. And cholera used to be regarded as a plague disease. That is, there were huge epidemics which would spread very, very widely and last for a long time, and were regarded as being a biblical plague in the old days. And but it's caused by an organism called Vibrio cholerae. I'll show you a picture here. Vibrio cholerae has, when you look at it under the microscope, it's gram-negative, and it often has this bent shape, even a U shape like this, sometimes a lazy spiral, just a single turn like that. And it has polar flagellation. And it's actually a very interesting organism because it's a normal part, especially of brack water, slightly salty waters, estuarine situations, and even in water which might be used for drinking, washing, even though it's not often particularly good quality. Again, this is one of these situations where there is a normal background population of the organism within normal systems. But under certain circumstances, certain ecological circumstances, the population of Vibrio cholerae can build up very high in the waters. And it can build up sufficiently high that it can cause an infection in somebody drinking that water. Now, once that happens, once you have one or few people who are infected with Vibrio cholerae, and they develop diarrheal disease, they are releasing huge quantities of bacteria. And if you have contamination of water sources by feces on people suffering from the disease, then you can end up setting up a situation where many, many people begin to acquire the disease. This is due to the disease process is due to a specific toxin, which is a cholera toxin, and it has the effect that it causes the intestinal walls to pour out fluid. And this is the diarrhea that results is so profound that people can actually dehydrate and die within hours. A child beginning the diarrheal process can die within a half an hour or 45 minutes. The body fluids simply flood out. And this throws all of the electrolyte balance of the body out. And the people die largely of the effect of the dehydration and then this complete disruption of their electrolyte balances. The characteristic after the initial flush of diarrhea is that people just produce a clear fluid. And it's called rice water stool because it's just a pearly gray look. It's just almost pure body fluid flooding out. An adult can lose up to 20 liters of fluid in a day. The disease is actually the presence of the bacterium is actually pretty well dealt with if you can provide supportive care. You don't even really have to provide antibiotics. If you can support with fluid support, IV fluid replacement, usually people will survive. Even in the absence of IV fluid replacement, if people can provide electrolyte-rich fluids, balanced electrolytes, and continually get people to drink, then they can also survive. It's definitely not a death sentence, but in the old days, it caused panic in a population when cholera arrived, because it spread, because people had poor sanitation. They were using pit latrines and this sort of thing, which often fed into the water source. Then this could spread extremely quickly. And the most famous story of all is one you've heard already, and it's the story of John Snow, the world's first epidemiologist, who during a cholera epidemic in London mapped out the households where people were sick and where they had died. And he realized that all of those households were drawing their water from one single pump, from a municipal pump. And he went to the authorities and said, this pump is the source of the disease. This in an age where they didn't really even believe that bacteria cause disease. But he went and he said, that pump is the source of this epidemic in this area of London. And if you don't control it, it's going to spread to other areas. And they wouldn't do anything about it. So he went at night, he stole the pump handle so that people couldn't draw water from that well. And he brought the epidemic to a halt. Cholera occurs very, very widely, but its mortality rate now is really, very low, provided adequate medical support is present. But you can see all of these dark shaded areas. It's actually regarded as being endemic to areas here in Pakistan and in Bangladesh, and then on either side at the north of India there. It's regarded as being endemic there, although quite honestly, you can isolate it from waters all over the world. It is particularly important and the area in times of catastrophe, because that is when there's a breakdown of things like the sewage system and support systems and all the rest of it. And very frequently following a war, refugee situations, earthquakes, these sort of things, they are followed by a cholera epidemic. And this is certainly the case in the latest, most disastrous outbreak, which was in Haiti. After the earthquake in Haiti, a group of, and very unfortunately, a group of United Nations support staff, soldiers actually from Nepal, imported cholera to Haiti, and it spread very rapidly. So in any disaster situation, they make preparations for dealing with cholera and with other diarrheal diseases. Not because the same things apply to other diseases. Oral rehydration solutions, IV equipment, etc. And then also immediately setting up the facilities so that people can observe proper sanitation, proper water storage, hand washing, etc., etc., etc. There is a vaccine for cholera, and it is used in disaster situations, but it's not a very good vaccine. It doesn't have a very high protection, very high success rate. It is worth administering, but it also is not very long lasting. If you do acquire immunity from it, it's not very long lasting, but it's worthwhile administering in disaster situations. Okay, so now, surprisingly enough, what we're going to talk about is our old friend, especially if you've been in a lab, you've seen this 100 times. And this is E. coli. And E. coli, Escherichia coli, a gram-negative bacterium. It's a facultative anaerobe, and it is a normal part, in fact, a major part of our bacterial flora, especially of our large intestine. And under normal circumstances, of course, it's completely harmless. We use E. coli as a marker organism for fecal contamination, not because we're particularly interested in a disease caused by E. coli, but because we know that most human beings maintain a relatively steady population of E. coli in their gut. And if we go out to a waterway, to a beach, or wherever, sample the water, and we find E. coli does not occur naturally free in water. If we find it in a waterway, it indicates to us that there is fecal contamination. And the amount of E. coli will tell us how much, give us an idea of how much fecal contamination there is. So when you hear, for example, that a beach is being closed because the E. coli, the coliform counts were too high, it means there has been some source of sewage contamination usually. The vast majority of E. coli are non-pathogenic and cause absolutely no problem. In fact, are beneficial to us. However, there are a number of different strains of E. coli. And it appears also that in different animals, some of the strains may be better tolerated than others, so that sometimes animals will serve as a reservoir of these types of E. coli, which are actually pathogenic. And E. coli, under certain circumstances and certain strains of E. coli, can indeed cause diarrheal disease. And these always pass fecal oral routes. And some of them, the disease caused is extremely mild, but in other cases, it's extremely serious. And actually, one of the most dangerous of all of our diarrheal diseases is caused by E. coli. So there are 5 major types that are recognized. The first is called enteropathogenic E. coli. And it actually causes a relatively mild diarrheal disease. It's very important, though, because it can be epidemic in nurseries. And where obviously very small infants have a limited immune range. And if exposed to this enteropathogenic E. coli, it can spread very rapidly through a nursery, and any diarrheal disease in an infant can be extremely serious. So it does something very strange, and that is here it is here. It actually adheres to the intestinal wall, and it causes the surface. This is a villus in the intestine. It causes the cell membrane to form these pedestals here with the bacterium attached to it there. That's characteristic of enteropathogenic. Apart from that, the disease is usually self-limiting. So the next one is called enterotoxigenic E. coli. And this is, again, one of these diseases associated with poor food handling and with poor hand hygiene. And this is often called traveler's diarrhea, because this is a kind of diarrhea which you might reasonably expect to pick up from street food or something like that. The symptoms are quite similar to that staphylococcal bacterium, and let's compare the two of them. Here are the differences. You're traveling and you buy food from a street vendor, and like 12 hours, let's say, no, not that, maybe not that long, 6 hours later, whatever, you begin to feel a bit ill. And then by the next morning, you have rampant diarrhea and you may be vomiting, etc. So what disease are you suffering from? Are you suffering from a disease caused by a toxin that has been released into food that you've eaten, or are you suffering from a disease caused by a living organism which has populated your gut? That span of time, 6, 12 hours, whatever, suggests that you are dealing with a living bacterium. You are more likely dealing with something like an enterotoxigenic E. coli. If, on the other hand, you buy your food from a street vendor and within half an hour you have violent diarrhea and vomiting, you are more likely dealing with something like staphylococcus. Why? Because what you're reacting to is a toxin. It's not the bacterium itself. The bacteria doesn't have to grow in your gut. You are reacting to a toxin which the bacteria produced while it was growing in the food. So that would be a difference between the two. This in enterotoxigenic E. coli, it's somewhat similar to the cholera toxin in that it causes the gut to flood body fluids, but it's not nearly as serious as a cholera toxin. And it is usually self-limiting. If the person begins to get dehydrated, they can usually deal with it by drinking more fluid. Okay, so we're kind of going up in intensity here. Enterotoxigenic E. coli, often called traveler's diarrhea, it's very, very common. And it's almost a characteristic of areas with poor hygiene, poor sanitation, where people are dealing with food and selling food under unhygienic conditions. Enteroinvasive E. coli. This is a far more serious E. coli infection, and it is usually acquired through fecal contamination, direct fecal contamination of food. Many of these really bad E. coli are acquired from things like slaughterhouses, areas like that, where meat has become contaminated with fecal matter from the animals that are being slaughtered. These E. coli tend to be, pathogenic E. coli tend to be well tolerated by cattle, for example, and can be acquired from cattle feces under various circumstances, either in a slaughterhouse. There have also been instances, for example, where people picked up apples in an orchard where they had been contaminated with cattle manure, and people then made cider or did whatever and acquired that way. In the case of Salmonella and of the E. coli, they can be killed by adequate cooking. And this is why it's very important to look when you are cooking meats, chicken, etc., to ensure that you reach a high enough temperature that you can kill off potentially pathogenic organisms like Salmonella and E. coli. So enteroinvasive E. coli is an E. coli strain which has some of the characteristics of Shigella, in that it causes an actual dysentery. It invades the intestinal walls using the same kind of mechanism that Shigella does, but it has this difference. Shigella also produces a range of toxins which cause illness. This one does not. It just causes illness by damaging the intestinal tissue. And it doesn't usually, it won't persist any further. It won't cause necrosis of the gut wall, but it does produce this very powerful inflammatory reaction. So it's in very painful, cramping diarrhea is one of the characteristics. That's enteroinvasive. The next one is rather uncommon. It's called enteroaggregative E. coli. It also is one of these E. coli that causes a strange reaction of the gut wall. And when you look at it microscopically, for some reason or other, the cells pack up with one another. They call it in like a fence or a palisade. This doesn't do a huge amount of damage to the gut wall. The problem with this one is it produces a toxin, and the toxin causes this watery diarrhea. It's an enterotoxin. So that means it just causes this generalized diarrhea and some nausea as well. So this, I've left the best for last. And this is enterohemorrhagic E. coli. This is the really, really dangerous one. Again, this is often acquired through direct fecal contamination of meat and things like that. And this particular strain of E. coli, the most famous one is 0157H7. That's its serovar label. And it produces toxins which are very, very similar to the Shiga toxins. And again, remember that the Shiga toxins, what happened was they were an endotoxin. That is that they were released as cells died. This is a gram-negative bacterium. It has an outer membrane with very powerfully immunogenic components that excite a very strong immune response. So the first thing is that this organism behaves very like Shigella, but it's not a Shigella, but it has the same kind of characteristics. It causes the hemorrhaging of the gut wall, and it causes a necrosis of the gut wall as well. And it can actually enter into the bloodstream. And when it does so, it produces the same hemolytic uremic syndrome that the Shigellas did. And one of the characteristics of this enterohemorrhagic E. coli, first of all, the hemorrhagic part is true, but there's tremendous amount of damage to the gut wall. But in addition, it sets off this situation of toxic shock throughout the body, of septic shock rather. And it causes widespread organ failure as a part of that septic shock syndrome. So this is a very, very dangerous E. coli infection. And this is one to remember, enterohemorrhagic E. coli. Okay, so those are some of the really more serious of the diseases. So this one here, I would separate this next one out a little bit because it's very unlikely that we acquire this bacterium as a result of the fecal oral route. May certainly be our initial exposure, maybe, but this is a bacterium called Helicobacter. And Helicobacter is interesting because it's one of the few bacteria which survive in the stomach. And it can actually be recovered from many people's stomachs who are not suffering any pathogenic effects whatsoever, where it is simply tolerated as part of the normal microflora. If you look throughout the world, about 50%, half people have some Helicobacter in their stomachs. The reason it's so interesting is it survives so well in that very low pH. And the reason for that is it produces this enzyme, which you've probably tested for in other bacteria, called urease. And urease breaks down urea, which is our principal nitrogenous byproduct, waste product. And it breaks down urea to release ammonia. And ammonia lowers the pH. So what these bacteria do is that they are creating areas of the stomach environment where the pH is kept low and they can survive and grow there really easily. However, overgrowth of the Helicobacter causes an inflammatory condition in the stomach wall. And that inflammatory condition disrupts the ability of the stomach wall to produce mucus and protective mechanisms which protect the gut wall itself from acid. Despite the fact that the tissues of the gut are producing the acid, which is released into the stomach, they themselves are susceptible to damage from the acid. So they are protected from the acid which they themselves release. They're protected by thick layers of mucus and various other substances. In an inflammatory situation caused by Helicobacter, those protective mechanisms fail, and the tissues of the stomach wall themselves begin to get eaten away, and that forms an ulcer, a stomach ulcer. This used to be attributed to things like worry and whatever. They used to sometimes regard people who had ulcers as being neurotic. It's got nothing to do with it whatsoever. Many of disease conditions which we suffer from are affected by stress. That is certainly true. But the cause of stomach ulcers is Helicobacter pylori, not worry. So, and it is treatable. It can be treated with antimicrobial drugs and various other things to relieve the symptoms of the ulcer and give the stomach wall time to repair. Okay, so here's the next one is another of these bacteria, which actually, in very, very low numbers, can be recovered from most people's guts in one form or another. And this is Clostridium difficile. Now, the Clostridia are a group of bacteria which are obligate anaerobes, and they are spore formers. They characteristically produce spores so that they can move through aerobic areas. They can survive aerobic areas only if they're in the spore form. They can't survive if they are in vegetative form. Now, the Clostridia have many characteristics, but one of the things about them is they tend to produce rather nasty toxins, and they cause very serious illnesses. Clostridium difficile is one which specifically causes disease of the gastrointestinal system. And it is very uncommon. It used to be very uncommon until heavy use of antibiotics began to be the norm. And the reason for that is that Clostridium difficile is very easily eliminated by a normal bacterial flora. It cannot survive, it doesn't compete well with other bacteria. And as long as you have a normal gut flora, you have no chance of establishing a Clostridium difficile population. But wipe that flora out with antibiotics, and Clostridium difficile finds it very easy to colonize the gut. And what it does is, here's a normal gut wall, but here's one which is being colonized by Clostridium difficile. And what it does is it induces the gut to produce a pseudomembrane. And underneath that pseudomembrane is anaerobic conditions under which the cells can reproduce. And they themselves cause an intense inflammatory reaction and cause ulceration and ultimately perforation of the gut wall. The gut wall tends to just become thinner and thinner and thinner, ulcerated, and eventually just break. So this is a potentially very dangerous situation. And this is very typical of people who have been on long-term antibiotic treatment, who have a reduced immune response, or who are very elderly. So, and in addition, this is a nosocomial, almost exclusively a nosocomial infection. Once populations of Clostridium difficile are established in a hospital setting, it's very, very difficult to get rid of them because they produce spores, which are liberated from feces and from people who are suffering from the illness. And the spores are almost impossible to clear up from all the hospital services, et cetera, et cetera. So you have a continual source of rich infection. A very, very difficult situation to deal with, especially in the elderly. It's not exclusively the elderly, but predominantly it is. So remember, I kind of skipped over the pseudomembrane story. I just want to remind you, remember we talked before about diphtheria. Diphtheria, Corynebacterium, did exactly the same kind of thing. It affects the cells, underlying cells, and it causes them to produce this extremely thick mucus, which traps all of the dying cells, all of the bacteria, et cetera, in a thick layer beneath which the bacteria can survive, free from interference by the immune system and everything else. The Clostridium difficile is extremely difficult to deal with, and very frequently fatal, especially in the elderly. But there is, for people who are suffering from chronic Clostridium difficile, the simple solution is to do a fecal transplant, to take some healthy feces from somebody, from a donor, and put them as high up in the large intestine as possible, and just leave them. And people have recorded positive results within days. Those bacteria begin to reproduce and very rapidly will populate the gut and exclude the Clostridium. Okay, so I'll just quickly mention to you a couple of the other bacterial diseases of note. The first is Listeria monocytogenes. This causes a disease called listeriosis. Listeriosis is potentially extremely serious disease. It's one of the more serious of the fecal-orally acquired diseases. Unfortunately, it is not that common, but periodically it does. You will hear of recall, for example, of meats, of cold meats, even of salads and things like this, because of Listeria. And the real difficulty with Listeria is, for most of our foodstuffs, it renders them really safe to keep them at low temperatures, because very few of the bacteria that I've mentioned are able to grow and reproduce at low temperatures. So if you refrigerate things, you keep the population, even if it's present, you keep the populations low. Listeria, unfortunately, it is one of bacterium that grows quite well at low temperatures. So it is a particular problem. There's another feature to it, which is a bit unusual, and that is that if a pregnant woman acquires Listeria, the Listeria can actually move across the placenta, and it can infect the fetus and cause spontaneous abortion. So that's another feature of it. It's a nasty, nasty bacterium. It behaves in some ways like Shigella. It can move into the bloodstream and can set up infections remote from the gut. For instance, it can cause a meningitis. It results in sepsis and septic shock, etc. So the next one is, I suppose, a little bit surprising that I had left it till last, but almost last. But this is called Campylobacter, and it's actually the leading cause of foodborne illness in the United States. And again, this is one of the bacteria which is commonly associated with fecal material in poultry and cattle. So we really tend to acquire it from poor meat management. And it causes more than 1 million cases a year, but self-limiting. It usually is a self-limiting disease, and it is really quite easily dealt with, even with over-the-counter medications. If it persists, if any of these diseases persist for more than a day or two, then they should at least be tested to see which organism you're dealing with, to know how to proceed. But Campylobacter really tends to be quite easily dealt with. The last one I'll mention is another of these picnic diseases, I suppose you could call it. And this is Bacillus cereus. And again, you would have dealt with Bacillus cereus in your lab. And Bacillus cereus is especially associated with food poisonings arising from grain. especially from rice, things like that, cooked rice, these kinds of things. If people have prepared a rice salad, for example, or sushi or anything like that, and have allowed it to become contaminated with this very common environmental bacterium, Bacillus cereus, which in general is completely non-pathogenic to us. You can swallow Bacillus cereus, and I doubt you'd ever establish an infection with it, but it produces an exotoxin. And if it's allowed to grow in rice, in cooked rice, these sort of things, it produces a disease very similar to the Staphylococcus aureus intoxication that we saw before. Okay, so we'll stop there, and our next one, we'll be discussing microbial diseases of the urinary and reproductive systems.